Hello, MCS teachers. Our very own biology teacher, Heather McNair, is keeping the wonder and curiosity going with three-dimensional lab demos. Her students are able to investigate and problem solve with their teacher from anywhere to build scientific skills needed for the next level of science rigor. Let's take a look at one of her lab demos to see how much fun you can have creating 5 to 10 minute lab investigations for your learners. As you will see, it doesn't take much to keep the investigations going during COVID. All you need is a recording device and your materials, and maybe a little bit of humor too. All right, let's take a look.
Hey guys, back with McNair again for another demo on surface area to volume ratio in cells, but we're going to be using these auger cubes. So auger cubes is kind of like Jell-O. If we were in class, you guys would be able to pick it up and feel it. It's a little bit harder, like maybe less squishy than some Jell-O, but we would put Petri dishes. This is the stuff we would fill Petri dishes with. So these are pink cubes. They have a dye called phenolphthalein in them. And when we mix phenolphthalein with either an acid or a base, then the phenolphthalein will either change color or it will kind of get bleached out. It will wash away. And so the cube becomes a little bit more clear. So we won't be able to see pink anymore. So what I have here is some vinegar solution in 4 beakers. And we're going to drop the cubes in there for the same amount of time to see diffusion rates. So how quickly the pink stuff gets out of the cubes based on surface area to volume ratio. So you can see here, because we're scientists, we measure in centimeters. This big one is a 3 centimeter cube. We've got this one as a 2 by 2 cube, and then this little baby guy is a 1 by 1 by 1 cube. Okay, so you should have already calculated the surface area to volume ratio of each of these cubes. And then this other guy back here is a 3 centimeter cube, just like the other one, except for I've sliced it and diced it. So same size cube, okay? It's still 3 by 3 by 3, except now so the volume is the same as this one, right? Except now I've cut that up into 27 little pieces. And so in terms of that, what I've done is increased the surface area. So we're going to drop these in, we're going to leave them in for 15 minutes, and then we're going to slice them open, and we're going to look at how much of the pink stuff actually diffused out. Alright, so let's check our results. I'm going to take them out by size. This is our big guy that I cut up, and it fell apart a little bit once I put it in the solution. Alright, so making some general observations. You can see in size again. The 3 centimeter cube definitely has quite a bit of phenolphthalein, so still pretty pink. Still pretty pink there too, but then you look at our little guy over here, and there's very, very, very little, if any, pink inside there. So view from up top. Alright, so that's our 3 by 3, and then what happened here with our 3 by 3 when I cut it up? Right, so I increased the surface area. You can see these are all smaller pieces. If I broke them all up here, okay, so they were cut into much smaller pieces. All right, and there's almost no pink there. So by increasing the surface area here, I was able to diffuse the same amount of more volume in the same amount of time. All right, so let's cut these open to see. Kind of our cross section there. So you can see we really didn't diffuse too far. If we look there, it's maybe, I don't know, 3 centimeters, 4 centimeters. Okay? We'll cut this guy open. And if we measure the diffusion again, oh my goodness, looky there, it's 1, 2, 3-ish. Sorry, millimeters, 3 millimeters, not centimeters, 3 millimeters. That's so weird, it's the same rate. Okay, same amount of diffusion. And then we're going to cut this little guy open. If he'll hold still, and I have no pink left. No measurable amount of pink there. So I think that's just kind of a shadow we're seeing, but no pink left there. So you can see there, that would have been that 3 millimeters, and that goes almost to the center there. So we have diffused all of the pink out of there, and so that's what we see here. So basically what I did here, by cutting this up, I did kind of like tic-tac-toe, and so I made these tiny little cubes. So you can see that we diffused all the pink out of there in the same amount of time, even though it had the same volume as this cube.
I hope you enjoyed watching Heather McNair's lab demo. If you are interested in receiving support to produce lab demos for your learners, or would like to view the corresponding lab demo handouts McNair created for her students, feel free to reach out. Thank you for spending a little time with your science coordinator today. And always remember to create opportunities for your students to ask questions, collaborate, create, make observations, analyze data, and communicate reasoning. I look forward to supporting you with science instruction. Take care.